Hello, Life Science aspirants. Welcome in the Life Science Academy. I am your Life Science educator, Neeta, and today we will focusing a highly recommended topic, that is the absorption in the PCT. And this portion is very important as for the CSIR NET examination, which have one questions from this part only. In this exam, one questions must, or you can say, coming from every year. So let's talk about absorption in PCT. PCT stands for proximal convoluted tubule. So as I already told you, nephron is the basic or functional unit of a kidney. It constitutes three parts. These three parts are the renal corpuscle. These three parts are the renal corpuscle, filtering components, and renal tubule. So what to do renal corpuscle? In the renal corpuscle, filter the blood to create filter the blood. What can happen in the renal corpuscle? Filter the blood to create filtrate. So to create filtrate or creating the filtrate. So in the corpuscle region, blood getting converting in blood getting converted into a filtrate, blood to filtrate formation, So and the filtering components and the renal tubule. What is the constituents of renal tubule? Renal tubule consists of PCT, leukophile, DCT, and collecting duct. These are the constituents of nephron which can participate in the absorption and as well as in the secretion. So simultaneously I will talking about the secretion also. So these are the three constituents which is responsible for the absorption and iron secretion and the collecting duct which is responsible for the final reabsorption of water and storing urine. So where the urine stored in the collecting duct So let's forward. The renal tubule can be divided into components: PCT, leukophile, DCT, and collecting duct. PCT has a high capacity reabsorption. This is the high capacity, or you can say vital capacity for the reabsorption. Maximum reabsorption can take place in this part. And in the part of DCT, if we talk about DCT, there is a conditional reabsorption. Some substances can be still present in the interstitium fluid. So if you want to further reabsorb, the further reabsorption can take place or in the DCT, or you can say distal convoluted tubule. This region is there is the presence of conditional reabsorption, not only the presence of urine. So it is, if we talk about the structure of PCT, So it is lined with simple cuboidal epithelium. If we talk about structure of PCT, structure of PCT, proximal convoluted tubule, it is lined with simple cuboidal epithelium. Cuboidal epithelium. It is lined with simple cuboidal epithelium, and this has this type of projection. This is the brush borders. This is the brush borders for which, for why? For increasing the surface area for maximum absorption. This is the surface area, to increase the surface area for maximum absorption. So this is the structure of the PCT. This is the structure of the PCT cells. Simple cuboidal epithelium which have brush border to increase surface area on the apical site. So this is the apical site, and this is the basolateral site, and this is the basolateral site. The site is the basolateral site. The epithelial cells have large amount of mitochondria. Large amount of mitochondria. So there is the presence of large amount of mitochondria present to support the process, involve the transport ions and substances. So moreover, they also have large number of channels. In this region there is presence of channel, in this region there is presence of channel, and in this region there is presence of channels on both the apical and basolateral membrane, which provide the large surface area for transport of ions and other substances to occur. So what is the functions of PCT? If we talk about functions of PCT, what to do done PCT? PCT done reabsorption. Reabsorptions of water, reabsorption of water, glucose, sodium, potassium, chloride, amino acids, and bicarbonate. So these are the substances which will reuptake with the help of proximal convoluted tubule. So in the PCT, this process occurs in the bulk transport, in the form of bulk transport. The solutes and water, remember, there is the presence of solutes and solvent. The presence of solute, these are the solutes, and the water move from the PCT to the interstitium, and then into the peritubular capillaries, So this part is the PCT, and this is the region, this is the interstitium region. So there is the presence of interstitium, and this is the peritubular blood capillary. Peritubular blood capillary. So as I can say, in the PCT, this process occurs in the bulk transport. This solute and water move from to PCT to the interstitium and into the peritubular capillary. So ultimate goal, what to do? Ultimate goal is via the interstitium fluid, these substances can move from this part to peritubular capillary, from this part to, or you can say tubular part to peritubular capillary. Peritubular capillary. This is the peritubular cap. This part to peri. So it's, for example, this is the part of the nephron and this is the part of the blood. So what we can say, what we want to, I want to transfer this glucose or this amino acid from this part to this part. This is called as the reabsorption. Reabsorption. This is called via the interstitium fluid. There is presence of interstitial fluid also. So I have taken glucose from there to there with the help of some transporter machinery, or you can say some transporter channels. The reabsorption in the proximal convoluted tubule is isoosmotic. So in the PCT, what will be the substances which will reabsorb? The substances are the substances or the, or we can say how the percentage reabsorption can take place. So 65% reabsorb water, sodium, potassium chloride and sodium, potassium and chloride, and 100% of glucose. Remember, 100% glucose is reabsorbed in the PCT part only, and 100% amino acid also, and 85 to 90% of bicarbonate and in bicarbonate is the main compound which play the main role in the balancing the buffer system of the blood. This reabsorption occurs due to the presence of channels on the basolateral facing the interstitium. This part is the basolateral part and this is the apical part facing the lumen, part. So there are two routes through which reabsorption can take place. The paracellular route, this will be the paracellular route, and this is the transcellular route. This is the trans. If the substances move, any substance move from this part to this part with the help of paracellular, and if the substances move from this part with the help of transcellular, this is the transcellular. Within the cytoplasm, there is the via the paracellular. There is the two route for the reabsorption of the substances, or the, you can say, glucose, amino acid, bicarbonate, sodium and potassium ion, etc. The driving force for reabsorption in the PCT is the sodium. So major driving force is what? Is the sodium. Sodium is the major driving force in the reabsorption of the PCT. Due to the presence of many sodium-linked transporters, for example, SGLT, sodium glucose linked transporter, on the apical membrane, So in the apical membrane, there is the presence of SGLT, sodium glucose linked transporter. On the apical membrane, sodium is usually co-transferred with solute, with other solute. Usually क्या होता है? Usually co-transported with the other solutes. For example, amino acid. For example, there is presence of some amino acids, and this glucose, there is presence of glucose, there is presence of sodium. So sodium, if you want to transport sodium, so sodium is co-transported with the amino acid also. The sodium moving down its concentration gradients, thus sodium moving down its concentration gradient to create an electrochemical gradient for sodium potassium ATPase. For sodium potassium, so sodium potassium ATPase, where present sodium potassium ATPase is present on the basolateral side. This is the sodium potassium ATPase. This is the sodium potassium ATPase, which take the ATP for driven the force. Examples: this is a type of pump, and pump has a facility. This leads to the 3 sodiums from the lumen, 3 sodium from the lumen, and as a contrasted 2 potassiums from the interstitium to the enterocyte or tubule lumen site. This transporter uses primarily active transport. This movement of sodium creates an electrochemical gradient favoring the movement of sodium into the cell from the tubule lumen. So already a gradient is made in the tubule lumen. So this gradient is used in the active transport. So there is presence of some co-transporters, refers to the movement of multiple solutes through the cell. There are 2 types of co-transporters, symporter and antiporter. The sodium concentration gradient allows other molecules such as glucose to be transported across the apical membrane against their concentration gradient. So these transporters, these co-transporter is present on the apical membrane. For example, SGLT. This is the example of co-transporter. Move glucose together with 2 sodium ions if there is the presence of glucose. And if we want to transfer this glucose in this region, so there is a presence of sodium ions. So there is presence of sodium ion, and this sodium ion, this is the co-transporter. This is a co-transporter. Then cross the basolateral membrane via facilitated diffusions. So nothing have to require energy for this. So via the facilitated diffusion. So if we want to transfer the glucose, from lumen site to the interstitial site, so with the help of facilitated diffusion, and this is a type of co-transporter. If we talk about sodium amino acid symporter, are present on the apical sites of the cells in the ascent segment of the PCT, which absorb all the amino acid in the PCT. So there is in the lumen side, there is presence of lots of amino acids also. If we want to reabsorb this amino acids, so there is a presence of sodium amino acid symporter, which will lead to transport the sodium as well as amino acids from the lumen to the interstitium fluid and ultimately to the peritubular capillary. So there is presence of antiporter also, sodium. So there is presence of antiporter of on the apical membrane. So this is the sodium and sodium proton antiporter. So antiporter, if the sodium move into the across the apical membrane, then the H plus constant H plus ion is moved into the lumen side, into the lumen to maintain the pH of the tubule. So the sodium ion move into the tubular side. So the sodium ions move into the tubular side, and while H plus ion is expelled out into the lumen, there is presence of bicarbonate also. And if we talk about bicarbonate ion, if the bicarbonate ion gradient is more in the enterocyte, so this will efflux in the interstitium fluid. As a result, chlorine ion is getting enter into the enterocyte to maintain the osmotic balance, So in this part, we will completing the absorption in the PCT. In next part, we will talking about the absorption in DCT, distal convoluted tubule, as well as in the collecting duct. So remember, in collecting duct, there is a conditional reabsorptions of the substances.